techniques in molecular biology course. And what I've done is just kind of made them more hardy for them to explore in 4-hour lab periods that might happen virtually for them either every other week or, depending on how the state of California goes, every week. So here we are. In a normal year, I have some bioinformatics where I take them to public databases and have them go through a workshop to explore the resources there. And then I also have some commercial software that I'm using, Snapgene, but there are certainly some freeware software out there as well. Serial Cloner is one of them that can do a lot of the same things that Snapgene does, but for free. And doing some primer design and molecular cloning there, enzyme-based and also Gibson assembly. And then finally, making sure that the students are taking this to heart and taking it seriously, I include this in my assessments. So they have take-home exams where I give them questions where if they don't know how to use the resources we explored virtually, it's going to be very difficult for them. So I'm trying to make sure that and reinforce that this is really part of being a molecular biologist, is knowing how to use these computational resources. And so that's kind of the setup of what I had and what I'll be doing in the fall and beefing that up. So coming up, NCBI is my go-to source for one of my workshops and having them explore that. And the other one that I use is the UCSC Genome Browser, is another great resource to get them to see 2 different ways that data is out there and that they can tap into. So in the bioinformatics workshop that I have, and I have a link for you guys so you can tap into a template of that workshop and adapt it for how you want to use it in your classrooms. But I usually start out by assigning them a gene. And that gene is usually tied to the independent project that we're going to do at the end of the semester. And selfishly, it's part of my research. And so I get them to go explore what I've been exploring in my own research group. And then at the end of the course, they're going to make constructs for me to use in my research lab. So it's something new that I need, and they get to figure out how to make it. So they explore that gene and they get into the gene database at NCBI, and they have to go through that full record and report out what's in there. What information they can find. And here's some examples, like thinking of the chromosomal record versus the messenger RNA versus the protein. Is it something that is validated, or is it something that's theoretical? Going into the single nucleotide polymorphisms databases, understanding validation, how many times it's been found, is it missense, etc. Links to GenBank and the different levels of that, reference sequences versus non-validated, and then isoforms or transcript variants that are present. So getting them to explore all of those aspects of it, and doing this via questions and having them find data, post things. We have an electronic notebook, so they do screen captures to show they've gone to certain places and found information, or addressing questions, or run a certain program or something and reporting out on that. And so this gives them the opportunity to, if we're in person, they'll be prepping for a lab and getting ready and finding information that's directly related to the lab they're going to do as part of an assessment, so their take-home exam. And then because I tie it to the gene they're going to work with at the end of the semester, it directly relates to the project that they're going to work on. And it allows me to kind of scaffold that information in there. So one thing I find is that defining the coding sequence, they haven't thought about that the messenger RNA file that they get is not actually the coding sequence. They have to find within that the actual coding sequence and get rid of the upstream and downstream elements, etc. And being able to find that in many places. So these are some of the things that I've done as the workshop. So here's a couple pages from the workshop I've set up. And if you go ahead to this website up here, our MCC Cures website, I've posted a Word document there as a template, and you can download that. And the orange highlighted text are the parts you'll have to replace for making it specific to what you want to do. But I've got some intro stuff ahead of time. I've got questions in green that they need to answer and post. And so if you want to, please adapt it to what you want, but share it back. I'd love to see what you guys do to make it better, or to explore different aspects of the databases that I don't. I have it very tailored to the information that I want my students to grab out of it for my course. So in my course, I have different experiments that they do. For instance, we do the lactase persistence laboratory that has been reported in BAMBED, but I don't give them the protocol. And so I tell them, we're going to go explore. Here's the chromosome location. I need 500 base pairs amplified in this region to get these 3 SNPs, design the primers. And so that calls upon all the bioinformatics stuff that they learned in the workshop, and they've got to then apply that to it. And then they use additional things to design their primers that we've gone over in our lecture part of the course. qPCR, if they understand what a chromosomal record is, they can then use the intron exon and figure out if the real-time PCR primers are going to distinguish between genomic versus messenger RNA in their sample. And so these are places where they can again apply all that information they're learning. And this really sets up the molecular cloning that I want to do in silico and in the lab. And so that's why I kind of threw this in at the front of this talk, is that it lays the foundation for me to be able to do that molecular cloning part in the lab. So the molecular cloning part, I use SnapGene. Again, I have a workshop that I have them go through to learn about this software. It's posted again as a Word document, and here I think I made things that you guys need to change if you want to adapt it in purple highlighted text if you want to do so. I have this workshop based upon malate dehydrogenase because we use that in our biochemistry laboratory, and that way they get to see constructs in their molecular techniques course that they will then again visit in their biochemistry laboratory, and trying to keep some consistency for the students. And so you'll see some of my questions are based on that. And in some instances, I give them files to start with. If you guys would like those files, please email me and I'm happy to share those with you as well. So SnapGene is the software that I use in the classroom. Advantages: they have lots of video tutorial support. A lot of the commercial websites where you're going to like Invitrogen or Addgene link directly to SnapGene as downloadable files they can load directly. So it's kind of nice to have that easy marriage between resources and the software. It's very intuitive, at least I find it so. Disadvantages: they do have a free viewer. You can download it and use it freely to look at different aspects. It does have some limited functionality in the free version though, so you can't do all the stuff in silico and exploring molecular biology that you could if you had the paid version. And of course, licenses cost money. So I emailed my chair last night and asked, like, how much did we spend per license last semester on that? Just to give you guys an idea. And so for our classes, they got a 1-year license instead of a 1 semester. So that was a change for us. And we had 18 students, but we bought 25 licenses because some of our research entities used it as well. And that cost us $2,000. So that was $80 per student if you break that down. Maybe you guys are better negotiators. You can maybe get that price down. But that's where we sat for this past year on that. And I know that they extended the license for us because of the COVID-19 situation. And so our students had it, I think, all the way through August 31st instead of ending May 15th due to the current situation. So different things there. A couple links here for some freeware. The Serial Cloner, it has a really good website. It does a lot of the same things that SnapGene does, but it's free. You can use it on Linux, Mac, and PC, and so it's cross-platform. APE, I don't know as well. I did use it in the past, but it's another option out there. I think I use that on Macs. I don't know about compatibility on other platforms. And so for SnapGene, what I use for getting into the cloning is kind of outlined here. So in their start, what they're really going to do throughout the workshop is create DNA files for me. And they're going to do it with different aspects. So we start by, I give them a GenBank ID number, they find it, defining open reading frames and adding annotations. I also make them go to Addgene and grab a vector and insert it into SnapGene and open it up. And in that case, they're going to annotate here. So here's a screenshot of that. So some annotations. They've labeled the gene. They've shown the translation. They have the his tag shown. They've laid down a primer and shown where it's going to be complementary to the sequence. And so this sets up them to see different aspects of the file. I then give them a vector plus gene construct in which they're going to do some sequence alignment. So getting a DNA sequencing result and aligning that to it, confirming site-directed mutagenesis, having practice with that, and then simulating agarose gels. So here's an example of the DNA sequencing result aligned. So the top sequence here is the native, the bottom sequence is the data. And in this case, it's a site-directed mutagenesis result that was actually generated by our biochemistry lab. So I grab up all of the data from the biochemistry lab when they make mutants, and I reuse it in the Multec as a way for them to explore data. So this is an example here of an alanine to glycine. I asked them to evaluate the data. You'll notice here that the sequencing is not awesome. So, you know, tell me, do you think this is a mutated file, and justify your answer, and getting them to evaluate the quality of the data. Here's an example of running a simulated agarose gel. So a restriction enzyme digest of 3 singles. So the first one, lanes 1 through 3 are single digests, and we have the triple digest in the 4th. So in class, I might do a restriction fragment puzzle where I give them a table of data and ask them to reconstruct a vector. And in this way, I could also simulate a gel and show them that, if that's part of kind of explorations that you do in your class. So this is one way where I marry that in and kind of prep them for if they're doing restriction enzyme digest in the lab. Here's a way for you to see your potential results before you get to lab, so that you know what you should see on your gel, and if you don't, how to troubleshoot and figure that out. And then finally, having them create the construct. So this is the in silico molecular cloning, where they have to go and find the donor vector that's going to give them a gene, and a target vector where they want to put their gene into perhaps a new expression plasmid. And I have them evaluate it for many different things. Can they do it just by cut and paste restriction enzyme cloning? So they can practice that in the SnapGene software. Are there compatible restriction enzyme sites that will allow me to make that transfer? And then they can do it in silico, and it will say, oh, well, your gene's going to be backwards. Or, oops, you cut out your GFP by using those sites. So yes, you can put your gene in, but you're going to get rid of your GFP tag. And so they can evaluate it that way, or design their Gibson assembly primers, run that in silico and see if it's going to work and amplify the way they thought. And so they can digest, ligate that together, and then look at the pedigree. And I love this one because I use this in my take-home exams. Once they've gone through and done this on the take-home exam, they show me their history of what pieces did they take. So here's an example of the vector, and they grabbed a gene and they cut it with Nico and Bagel, and then they ligated it together to make the new construct. And this is an easy way to evaluate what they're doing, but it's also an easy way in presentations at the end of the semester on their projects to show me the route they took to make their construct that everyone can follow. So here's what my semester is going to look like starting August 17th. We are in the hybrid mode. So in my class, I have one lecture that's about 80 minutes, and then I have a laboratory that's 4 hours once per week. Because of social distancing in the lab, I can only see half my students. So I've divided them into in-person 1 week and virtual the following week. And they'll rotate between those phases. We have 6 modules: basic skills, nucleic acid manipulation and PCR, and then more advanced skills, advanced PCR like real-time nucleic acid manipulation like CRISPR, which Celeste will probably talk about at length in hers, and then independent projects. And in person in the laboratories, you'll see I have it broken down into those kind of experiments. And then virtually, I talked to you today about the bioinformatics SnapGene. If you do those 2 blocks together, it's about 4 hours, and the students have to mop up a little bit of their files on their lab notebooks, but it's about a 4-hour exploration to go through, so that will fill that time. We also do a standard operating procedure where students write up. I give them a template for a company, and they prepare one of those and do a peer review on that. So that's one of my virtual labs. Primer design is really big. It's amazing how long they take doing that. So complementing lecture and doing that, that's part of my virtual project design for what we'll be making this semester. And then there are some great case studies that came out at the National Center for the Case Studies on COVID. There's 3 or 4 of them. And my colleagues in biology who taught this last semester took 3 of those, modified them, and used them in Moltec to explore the evolution of the virus and different aspects of the testing. For instance, when they screwed up the qPCR primers, why was that such a big snafu, and how you could have caught that, etc. And so those are available through the national website to do those. And if you want to see how we modified them, I'm sure that we'll be able to share those as well. And so that's a really brief overview of kind of what's going to go on in my life and things that I've adapted, and hopefully some resources that you guys could use and help me make even better. And with that, I'd like to thank all the faculty at USD who helped make the biochemistry program awesome, especially my students who are fabulous beta testers for all the crazy ideas that I have, and they're very gung-ho about them. And of course, ASBMB for giving us this forum to share some of the things that we're doing and get better at it. So thanks, guys.
Jessica, thank you. Couple questions that came up. One is about, do you use electronic notebooks in your labs?
I do. I use Lab Archives.
And you like that?
Yeah, and we use them from General Chemistry just started, OChem uses them, and now Biochemistry and Moltec. We've been doing it for about 3 years.
And just out of curiosity, have you ever tried to run this lab with and without the SnapGene to see if they have a better understanding of kind of the processes and the techniques?
Yes, the first 2 years, first year I ran it without SnapGene, really hard. We did a lot of stuff on paper, which they don't like, or cutting and pasting in Microsoft Word. There's freeware called BioEdit that I use. It's only good on PCs though. And I tried to show them in that, but the Mac people were quite upset with me. We do have some departmental laptops that are PC that I borrowed out to them, but then of course they can't do it at home. And so those are some limitations that I had.